Hi, my name is Professor Joey Wallace. I'm the chair of the Biomedical Engineering Department here at IUPUI. I'm also the director of the Bone Biology and Mechanics Laboratory. As the name of the lab implies, our research focuses heavily on bone mechanics, or the ability of bone to bear external loading, and mechanobiology, or how cells and tissues respond to mechanical stimuli to maintain or increase bone mass. As opposed to being a static and inert support system like the girders in a wing or the frame of a building, bone has the unique ability to reshape itself to accommodate the ever-changing needs of our body. Our lab uses this characteristic of bone as a possible therapeutic by studying combinatorial treatment options for a number of relevant diseases which have musculoskeletal complications leading to bone fragility and fracture. These include diseases specifically of bone, such as osteogenesis imperfecta, as well as more common diseases such as diabetes and chronic kidney diseases, which also has skeletal manifestations. We utilize techniques and tools that are common in material science to study bone quality and fracture resistance from an engineering perspective. Doing so in collaboration with bone biologists and clinicians at IUPUI and the IU School of Medicine and beyond makes this a very powerful approach. Our lab includes postdoctoral researchers who have already earned their terminal degrees, as well as PhD and master's students. Of particular importance is the value we place on undergraduate research and the hands-on, real-world lab experience that they gain as they move through our BME undergraduate curriculum. Two current PhD students will spend a few minutes introducing some of the in vitro and in vivo models and methods we use in the lab.
I'm one of the PhD students in the lab, and I'm going to give a brief overview of the mechanical analysis that we do. So this is the mechanical tester. It's one of the main workhorses of our lab, and we use it for two main things. First, we use it for mechanical characterization. We'll put a bone in here, as you see, in either a 3-point bending setup like here or a 4-point bending setup, and we will apply a displacement load where this mover head moves down until the bone breaks. And when the bone breaks, the output data that we get from that gives us a lot of important information, such as how strong the bone is, how tough it is, how the bone material can resist a crack moving through it, all kind of data that allows us to quantify whether a treatment improves bone strength. The second thing we'll use the tester for is as a treatment itself. So we will put a live anesthetized animal in here and apply a cyclic up and down load that mimics an exercise load, which in turn increases bone mass, which can improve bone strength. So the two main things we use the tester for are analyzing the strength of bones and as a treatment to increase the bone mass.
Hi, my name is JD, and I'm one of the graduate students in Dr. Wallace's lab in the MD-PhD program. To help complement our in vivo studies in the lab, we also do in vitro studies using cell models of bone cells. We do these studies on osteoblasts, which are the main bone-forming cells, as well as osteocytes, which are thought to play a really important role in mediating the mechanical signaling of bone, as well as controlling the actions of the other cell types within the bone. One way that we study this is using mechanical stimulation of these cells using a flex cell device. In this device, cells are plated onto a 6-well plate with flexible membranes on the bottom. By using air pressure through these tubes, the bottom of the dish is deformed, which stretches the cells. We can then collect the cells and analyze how they respond to mechanical stretching. Other ways that we do this in the lab are through parallel plate flow chambers, which subject fluid shear stress to the bones, which is another type of mechanical force that the bone can experience. Our overall goal with these projects is to understand how osteoblasts and osteocytes work together to build, alter, and remove bone in order to improve bone integrity.
For more information, you can always check out our website at bbml.et.iupui.edu.